I'm Thornton Larson, Dr. Thornton Larson from Sulross State University, and this is STEM Paths, where students unpack papers, study paths, and the people behind them. Our guest hosts today are Ariana and Destiny, and they're going to be interviewing Dr. Jinchuan Xing from I lost my view. Today on his paper analysis of DNA variants in microRNAs and microRNA 3' UTR binding sites in female infertility patients. So if you guys want to take it away from here, feel free to go.
Yes. And so Dr. Xing, it's very nice to meet you. My name is Destiny Acosta. I'm a senior here at Sulross State. And my major is in biology with a concentration in pre-health. I'm looking into getting into the dental field, so hopefully I'm successful in that. But we're excited to review your article with you and ask you a couple questions on the analysis of DNA variants and miRNAs and miRNAs 3' binding sites in female infertility patients.
And hi, Dr. Xing. I'm Ariana Escalante, and I am a senior here at Sulross State University. And I'm on the path of obtaining a Bachelor's of Science in Biology with a concentration in chemistry. So, like Destiny mentioned, we are very excited to have the opportunity to speak with you today regarding your analysis in the field of infertility. So to begin, we can acknowledge that infertility is a dominant problem amongst women, being seen in predominantly 8.8% of women. To begin discussing your paper, we know you and your team evaluated existing data that is available, acknowledging whether regulatory glitches in these specific maternal genomes can be an explanation to explain the recurrent embryonic arrest that patients may experience. And what you guys noted in your paper was the specific and important transition period of the maternal to zygotic transition. And this is a crucial component that occurs after fertilization and day 3 of development, in which the cell needs to ideally become its own power plant and become independent from the maternal genome. So if these maternal instructions aren't cleared properly, right, or the embryo's own genes don't activate successfully, development ultimately stops. And through evaluating your paper, we see that you and your team hypothesized that in some cases of embryonic arrest and pregnancy loss may tentatively be explained through mutations found in the maternal genome, which ideally then translate to affect miRNA target transcript pairs. So you and your team initially looked at normal embryos and compared these to the population that was considered high rates for aneuploidy. But you and your team decided to take a shift as there was unfortunately no statistical significance between these two groups. So we proceeded at least in your work too, that you guys analyzed the mutations found in the seed regions and then those in the major landing pad of the 3' UTR binding site. And specifically what you guys found in these 3' UTR mutations was 2 major variants that we see were just really significant, showing 40 times more common, being more common in the infertility patient cohort when compared to that normal population. And this is roughly 1/3 and 1/4 of that population you guys evaluated. So as we can see, it is pretty significant. And just the takeaways from your work to summarize in short is that genetics disrupt maternal transcript clearance, but this data can ideally be used as potential biomarkers that can be implemented when screening patients, and specifically maybe evaluating those that may be at a higher risk for infertility. And just to continue on, these ideally may lead to potential targeted therapies, which is exciting, Dr. Xing.
So with that, Dr. Xing, did we miss anything else?
You gave a really nice summary.
A lot of key points, a fantastic paper to break down.
Okay, so going on to our first question we have for you, we would like to ask you, did you look at whether these variants differ between primary infertility and second infertility patients? So those who haven't had a successful pregnancy compared to those who have had one but have been unsuccessful the other times?
The short answer is no. One thing that is difficult in this kind of research setting is we can only work with what we have, and recruiting patients is on one hand fairly difficult. On the other hand, we don't get all the phenotype data. So in this particular collection, what we have is their PGTA, the pre-implantation genetic testing result, right? So we know under the IVF procedure, how many oocytes from a particular woman is aneuploid. But we don't know their medical history or their pregnancy outcome even, because that's not part of the study design. We are not allowed to have that information. So that is an interesting angle to look into. Unfortunately, for this data set, we don't have the phenotypic data to separate the people into groups.
Yeah.
Thank you so much. You pretty much summed it up for us.
And yeah, I could see how that may be limiting, not being able to have the access to that medical history of these patients. But I guess from your experience right now, Dr. Xing, if you had to, I guess, assert a hypothesis in this as to what these differences may look like between these two groups, would you consider one maybe being higher than the other?
Yeah. I would say it depends on the genes.
Okay.
So what we are looking at really, if you think about fertility and aneuploidy risk, we are not looking at infertility. So these people come to IVF clinic, and some of the women have higher risk of low quality eggs, but most of them having kids eventually. Right.
Okay.
So we are not, if you think about genetics, we are not looking at the genes that's basically knock out the function of reproduction and cause infertile. So we have a reduced function. That's why also this kind of regulatory miRNAs are important, right? We are modulating the protein's expression level rather than say the protein is losing function or not.
Yeah.
Right. So in this case, the mutations we find will confer a range of risk. Right. So some of these people might having kids and just having difficulty have kids in general, or they have a more severe case, you really need to have IVF procedure for them to have kids. So again, when we look for risk variance, our expectation is some of these mutations going to have either a little more dramatic impact or less dramatic impact, right?
Yeah.
So it's hard to say if you're going to find things that's only in one category versus the other.
No, well, thank you for your comments in that regards to that question. So to continue proceeding, we really loved the aspects of your paper of choosing to include the figures specifically of the regions in which the mutations were found in the microRNA. So random question, we were just wondering if diagramming that specifically was a goal for you and your team to kind of help visually articulate that information to your audience of where these mutations might be found in microRNA.
Yes, that's exactly the goal, because this journal have a primary reader of clinicians, right? So not everybody is in the genomic field think about microRNAs every day. And the microRNA have a very particular structure, and different components have very different functional role. So we feel it's important to provide a diagram to give people an intuitive sense. When we talk about seed mutations, we talk about loop mutations, where are they in terms of the microRNA structure?
Yeah.
Really quickly, is there a difference in writing style you put out for papers when you're writing for clinicians versus other researchers?
A little bit, not by much, right? I would say more generally, if we are writing a paper for a multidisciplinary journal, let's say Nature or PLOS ONE, right? When we know the reader is going to be very widely distributed in terms of their expertise, we try to make the introduction more general. We also try to make the discussion more general, but the core component is going to be scientific. It's not going to change much. But if you are submitting to a very specialized journal, say genome research, then the assumption is the readers probably have a very basic sense of genomic knowledge. So the introduction and discussion going to be a little more specific.
Right, absolutely.
Yeah. Okay, so I have another question. What were the initial motives that led to such a vital research in the field of infertility?
Right. So this is Collaboration. So my lab is a computational lab, right? We do candidate gene identification, we do genomic analysis. My collaborator, Dr. Karen Schindler, she's a cell biologist and geneticist, and she had a long-standing interest in egg development and nucleity in the egg. And then in our team, there's also collaborators from the IVF clinic who had the DNA bank that provided us the opportunity to look at the human data. So it's just how the project came together is you need all three pieces for this project to happen, right? Then after communicating once everything is in place, we developed this project with sample with people who had expertise to do analysis and with people can do functional validation once we have candidates. So it's a question that's important for sure. But in this particular case, it's just all the pieces falling in place around the right time.
Yeah, it shows it.
How we started it.
Yeah, it really takes a team to conduct research and, like you said, piece it all together. So with that, kind of to speak on again the data analysis, do you feel what were the personal challenges you believe you had with the whole exome sequencing? I guess working with that type of data.
Right. So I don't know how much you guys learned about whole exome sequencing. So this is sequencing.
We haven't touched on it much yet.
Yeah.
Okay. The ideal situation is you do whole genome sequencing, right? You sequence the entire genome, you get all the variations in the whole genome. Whole exome sequencing is you are particularly enriched mostly for the protein coding sequences. So you are sequencing 1 to 2% of the genome instead of the whole genome.
Yeah.
Which is much cheaper and much faster, easier to deal with. So it's kind of an intermediate stage that nowadays a lot of people use before they move on to whole genome. So it is easier to analyze, but on the flip side, you are losing all the regulatory elements because you didn't sequence 98% of the genome that's not protein coding.
Yeah, only 2%, right? Is like that coding region.
Yeah. So that means we are limited in terms of what kind of mutation we can look into. Right. And in this case, we have microRNA gene that's part of the exome sequencing. We have the 3' UTR of protein coding genes that we can look at. But things beyond that, it is pretty certain there are other regulatory mutations is going to contribute to the phenotype. But in this technology, we are limited to, we just couldn't look at those. So it is a discovery project. So we discover what we can find based on the data we have, but we are not looking at the whole picture.
Okay. And if it's possible, if you can go into detail for us, what did the writing process look like? And did you and your team run into great challenges during the publishing process?
The writing process, typically, I have the first author is the person that's doing most of the analysis, who is most familiar with the data. So typically the first author and me will be drafting the manuscript together. Mostly the first author will do the initial draft, and then we lay out the results section first to say, okay, what are the main messages? What are the main findings? And then we lay out the display items, the figures, the tables, and so forth. And then we start putting in the details. And in that process, all the co-authors will kind of responsible for their corresponding part. Right? The people that involved more in the sample collection, for example, will write a master section about the sample collection. And people that's involved more in the downstream analysis will write a specific section for the analysis. And then we kind of, but the primary responsibility is on the first author and me. So we kind of synthesize it, polish it, and make it into the final form for submission.
Yeah, the final product, which I'm sure, again, takes always a lot of work, and it's a process getting there.
It is a process.
Yeah.
And
So
for this particular paper, the publication was pretty simple because we are submitting to a special issue of the journal. So they are particularly looking for computational analysis under the clinical setting. So we have the journal interest lined up already before we even submit it. So they know what we are doing. They are interested. So the review process was pretty efficient, I would say.
Quick question: was the first author on this paper, was it a student of yours or more of a colleague? Because you're the corresponding author on it, but I just want to ask for the sakes of these students here.
Yeah, the first author was a postdoc.
A postdoc. Okay.
She started her own lab now. She's
that's so
assistant
cool.
professor now. Yeah. But she was training as a postdoc with me, so she's very capable of doing the analysis and writing the paper. Yeah.
Oh, that's awesome. So to transition now, Dr. Xing, what was the most rewarding part of conducting this study and being in the way we're discussing at the finish line and completing the publishing process?
Right. The most rewarding part for me is the potential clinical connection.
Yeah.
Right. Because I enjoy doing discovery. Right. I work with genomic data because it allows me to find new things. But for most of my project, we focus on the basic underlying biological mechanism. Right. It is very satisfying to understand how the process works, and down the line we'll get drug discovery, but it's a long process. Right. Usually from what I found as a candidate gene to a drug that can be used in the hospital is probably going to take decades.
Yeah.
But this, if we get things that have strong evidence, we can see how this can be used as a screening or doing prognosis in patients, and they can have a real world implication and impact much sooner. So I think that's one part that's really important for this is if we find something, for example, the candidate variant that's much more common in our patient cohort compared to the general public, that the information can be used without knowing all the underlying biology.
Yeah.
Right? We can use it as a marker.
Yeah, biomarker. Yep. So to transition, Dr. Xing, like you said, we have your paper uncovered really important and significant findings. Over time and within these last 5 years, have you developed preferred methods to articulate that knowledge and information in the field?
We have not going much further at the moment. What we are looking around actively is recruiting a bigger cohort.
Okay.
Right. So one of the limitations of our study is we only have a couple hundred patients. So the signal is strong, but we need to make sure it replicates in a much larger cohort before pursuing it as a biomarker.
Okay.
And as I mentioned, getting both recruitment of the patient, also the access to their clinical phenotypes is time consuming. So we are still working on that and try to get more people so we can validate the results.
Since infertility is kind of a sensitive subject to certain people, does that make it more challenging to obtain that data? They're less willing to share that data because of some of the stigma that comes along with infertility?
I think it's a part of it, although we are working with IVF clinics, so everybody go there are kind of having, trying to have kids through the technology help.
Yeah.
So they are, as a cohort, in general, more willing to participate. The issue is for IVF clinic doing genetic research is not part of their day job. Right. So the infrastructure of getting informed consent, getting IRB approval, all the proper regulatory procedures are not easy for them to establish. Right. So we are lucky to have the collaborator to start initially, and they have established a biobank of several thousand patients before they started doing the sequencing study. But we kind of exhausted samples in that collection. Now recruiting new patients, even with the infrastructure in place, is going to take time.
Absolutely.
Right? Yeah.
Can you elaborate on the impacts you and your team initially hoped this research would have? Now being 5 years from the publishing date, do you feel it was accomplished?
Accomplished is a big word. I think I wouldn't say it's accomplished, but I would say we are getting closer to our goal. So for this infertility or difficulty in conceiving, there is a real world implication and a very actionable thing to do, because the woman's risk of aneuploidy increase with age.
Yeah.
Right. So age is a major risk factor. And if you have something that's predictable and we can test people when they are young, they could be still normal or could be still have a much lower risk if they don't have ability to or opportunity to have children right away. They could also consider either freezing eggs or there's alternative approach, right? So even without the drug, I think our results can have a direct impact on how people make decisions in terms of reproduction if we can have good predictive power. So I think that's really the big goal we are shooting for. It's not necessarily in the end, quote unquote, treat infertility, right? That's a longer term goal. But if you can provide prognosis when women are young, I think that can help people much more directly than having a drug down the line.
Yes. I believe kind of going back to earlier, it helps manage patient expectations. And like you said, at a younger age, kind of just giving them that information that would allow them to be proactive and hopefully conceiving in the future if that is their goal, and would be at risk for, unfortunately, not being able to be successful. So that was awesome.
Exactly. Because I think for people, nowadays, the reproductive age is getting older because education, career, and everything in life. But if you know you're going to have a higher risk and you might have trouble when you are 35, you might, alternate your life priorities and do something. So I think the information is powerful.
Yes, very. And if I could comment quickly on that. So I'm aspiring to pursue the medical field. So ideally the goal, 4 years of medical school, proceeding to residency. And I'm like, now these are raising questions and concerns for myself where I'm like, where would I be along that timeline? Because like you said, career demands and education, it does shift your priorities and availability really to be able to prioritize that family aspect of being able to be successful, to have offspring again, if that's a goal. So it's definitely raising ideas in my head and important things to start thinking about. And of course, when the age is right, but I'm an important biomarker for sure.
Now transitioning, can you walk us through how you got to your current position?
Okay. So I started my undergrad as a biochemistry major. Okay. So I'm interested in biology all along. And initially, I think I'm going to really work in the lab and testing particular hypotheses, understanding the pathways, supporting interactions, and so forth. And during my training in PhD, I started getting interested in evolutionary biology and genomic science, I would say. And that's the time when the human reference genome just got published. Now, all of a sudden, we have this entire genome and have all these questions that can be answered. That before, people only have the question and they have no tools to answer them.
No resources.
So I started learning genomic analysis. And then during my postdoc, the sequencing and other high throughput technology in generating genetic data is getting quickly adapted. All of a sudden, I realized I have so much data that I don't need time to generate data anymore. I need to analyze them. So I kind of transitioned to computational analysis because there are just so many questions that can be answered with the data that's already available. So I kept a small amount of wet lab components when I started my independent position at Rutgers. But the lab is heavily switched towards computational analysis. And over the years, it switched entirely to computational analysis. Now, all my students are just doing discoveries using the data set, either we generated ourselves or from the public resources. I think nowadays, generating genomic data is not a hurdle anymore. It's getting cheap and easy to do. The interesting and the hard part is how you identify signals within the large amount of data that you start with.
Yeah, the interpretation aspect of all the data that is accessible. So then to, I guess, continue on this path, how can we as college students work to getting to where you are at right now in this field?
Well, I would say one thing is get into research early and get a sense if this is something you want to do. Research is a very interesting career option, I would say. But they also have a lot of struggles, right? I wouldn't say it's for everyone, but if people are enjoying it, they really enjoy it. Right?
Yeah.
So I think if you can start early, which allows you to get maybe not even longer, but also rather diverse research experience. Because again, for research, if you are working in the lab or working on the computer or working with model organisms, is very different experience, right? So having experience will help you to decide if that's something you want to do. The other thing is, I would say, follow closely at AI at the moment, because I think the AI is going to have a really big impact on what we do very quickly. It's already happening, but I imagine it's going to be much more involved in the next few years, definitely in your guys' career. So figure out how it can be used and how that is going to be part of the research is going to be important for your career.
Yes, very. Yeah, we definitely see that side of aspect being in college and seeing how it just continues to advance, and a lot of positive ways, of course, that it could be implemented. But like you said, consider how it can affect your career position or what you're choosing to pursue. With that, I had a quick question going back to you discussing the different realms of research and kind of your positions. Did you have a personal favorite? Has it been like getting down and working with the genome or the analysis portion strictly, or any specifics?
Yeah, so I consider myself a biologist at heart.
Okay.
Right? And I'm not, getting on a boat and go observing fishes now, but the genome is really my playing ground. Right? Well, every time I get a data set with a number of people, or even I also work with non-human species, I just get a data set of a bunch of genomes. That's the most exciting part, right? I don't know what I'm going to find.
Yeah.
I will go in there and try to figure out what are the discoveries. And this part of discovery is for me, is the most exciting part of my job. So I enjoy the aspect of discovery, and I want to keep doing that.
Yeah, definitely something always you can look forward to every day. You don't know what you're going to find, but I'm sure there's
Yes.
a lot of, we know the human genome, there's a lot that can happen and a lot of opportunities for, like we evaluated within your paper, just mutations in all these regions in which they can occur. So I'm sure it makes it a definitely exciting aspect of your day-to-day life and job.
Yeah.
Quick question.
I love, but to be surprised, right?
Yeah.
And often I can have a working hypothesis in my mind when I start doing the analysis. Often it doesn't follow.
Yeah.
I usually find something that was not what I expected, and that's exciting.
Yeah.
Quick question. I'm so sorry. When looking at conducting the research, could you tell us a little bit about the expenses part of it? Like, do you spend, how does it work?
Okay, so for computational research is much simpler than lab setting, right? Most of my research costs actually go to personnel. I have to, support my students, my postdocs, my technicians, so they can do the work. I have a lab server that people work on it, which, once we purchase the server, the computing is free. And we have a large university cluster. Again, most of the time it's shared resources from the university, so we can just do computing there. The only time I have to pay for computing is if you have to do cloud computing. If you have to work with Amazon Cloud or Google Cloud, then there is a cost associated with the computing and data storage. My lab don't have a heavy footprint in cloud computing, so it's not a lot of money compared to personnel cost. That's going to be very different. Say for my colleague work with mouse colony, her cost will have a very large footprint in reagent cost and mouse facility and so forth.
Okay. Thank you.
And so, Dr. Xing, what do you believe are some of the largest challenges that are facing scientists who are completing research in this field of infertility and working with IVF patients?
I would say sample recruitment is the largest challenge, because we not only we need discovery cohort, right? We need to have patients that we can sequence, we can get results. We also need large replication cohort to show that whatever we found are real, and it is applicable to a large population. But if I'm just doing discovery of mutation that cause particular phenotype, let's see, I have the mutation, it's good. I'm going to go to the library and do functional testing. But if you want to develop markers to say this is applicable, a mutation that's disease causing but only show up in 1 out of 1 million people is not going to be a useful test. Yeah. Right? We need to find markers that's common enough in the population so it makes sense to screen people with it. So to do that, we need to test a lot of people. The tricky thing now is the IVF system, OBGYN is separated. So once, say, a couple went through IVF procedure and the mom is pregnant, they move out to OBGYN and there's no connection anymore.
Oh, wow.
So the IVF clinic doesn't even know the pregnancy outcome.
Yeah, whether they're successful or not.
Yeah, so there's this separation. So whatever we get from IVF, we cannot really map to the OBGYN results. Whatever we can get sample collection from OBGYN, we don't really know what happens before that, right? So that makes the collecting samples challenging.
Yeah, and I'm like, maybe now.
We are working on ways to try to get around it. Now there are a lot of large collection of cohorts that's not phenotype specific. So we are tapping into them. Let's see if we can get enough information for people's pregnancy outcome and so forth.
Yeah, and it definitely shows the opportunities for potential working connections between maybe, like you said, those IVF centers and then communication within their OB to show whether they were successful at the end. I think that's an important aspect to look into, and definitely an area that can continue to be progressive in this field to help overall the data findings that continue to be put forward.
Well, Dr. Xing, thank you so much for your time and for the vital information you shared with us today. 3 quick questions for you. What is one piece of advice you would give to your undergrad self?
Keep pursuing your interest. I think, I've been enjoying research starting young, and I just stayed at it. I think it's, for me, it's a fascinating career.
Yeah.
And if you don't mind me asking, how old are you once you feel you kind of stepped into that water and realm of research?
I would say high school.
Oh, wow.
Roughly. Yeah. We had some chance to interact with scientists during high school. And then I started going to the lab, doing research second part of my college.
Okay.
And there I get a better sense what research is like, and then just still enjoying it and just kept pursuing this direction.
And do you believe these were opportunities you were continuously seeking, or do you believe just kind of you had well mentors along the way who were maybe kind of trying to encourage your participation in the field or just research in general?
The mentors are crucial. Mm-hmm. I think I always had the interest, but the mentors are crucial. Because this is exciting career, but it's also a hard career.
Yeah.
I think that along the way we need a lot of support. And I am lucky to have good mentors that provide encouragement, support, education, and that really allowed me to get to where I am. Yeah.
That's awesome.
And if you weren't doing this, what would you be doing?
If I weren't doing this, I think I would still like to do research. If I'm not in university setting, I can imagine in a research institute or even in industry. Because, large industry companies now, they also have large research and development R&D components. So I have a number of students now are working in industry, but their work is still very much similar to what I'm doing. But they come up with a hypothesis, collect data, doing analysis, doing discovery. So,
Well, thank you for that. So for the last question to wrap up our interview with you today, Dr. Xing, what's one thing you wish more people understood about science or scientists?
we are also people.
Fair.
I think when you say scientist, it sounds like a big word, but really is we are the same people doing day-to-day life. But I think the scientists are a group of people that have intense curiosity, right? We are not satisfied with working the same job every day. Even it's scary and risky, we want to find something new every day.
Yeah.
Yeah.
Which, like you said, again, makes every day exciting. And that's how we're all continuing just to advance forward in this life with all the research findings that you guys, of course, are doing all the hard work in. So we thank you so much, Dr. Xing, for your time today and all the knowledge you poured into us. We learned a lot.
Yeah.
Thank you
With-
so much.
Yeah, with that, I'd like to make sure and thank Dr. Jinchuan Xing of Rutgers University for being our guest today, and thank Ariana and Destiny for being such fantastic guest stars. This has been STEM Paths, where we start with a paper and we bridge into some bigger picture stuff and the researchers behind them. So thank you, everybody, for coming out today, and thank you again. Yeah.
Thank you.
Thank you,
All right. Thank you.